One aspect of muscle physiology that's received considerable attention since the late 60s and early 70s, when investigators started sampling pieces of human muscle using the biopsy technique, was the classification of muscle fibre types and their relevance to exercise performance. The muscle biopsy technique was introduced by some Scandinavian researchers to exercise physiology research in the late 60s. It involves taking a small piece of muscle using a specialised needle under local anaesthesia. In this case, you can see the needle inserted into a muscle of the lower leg to extract a small piece of muscle. And you can see in this picture here, the muscle being removed from the needle. This piece of muscle can be mounted in special chemicals and analysed for histochemistry. It can be frozen and later analysed biochemically or using immunoblots to characterise the protein concentrations of various key enzymes. This technique is widely used in exercise physiology research today to obtain pieces of muscle as part of research programs examining a whole range of aspects of exercise effects on muscle. In relation to muscle fibre types, there are two main categories, slow twitch and fast twitch. The fast twitch fibres, in human muscle at least, can be further subdivided into two subcategories, and we'll come back to that in a moment. The traditional way of typing these muscles is to mount a piece of muscle obtained from a muscle biopsy in a special medium, to then cut it in a cryostat, and then to stain for an important enzyme in muscle, the myosin ATPase. The activity of this enzyme is differentially sensitive to acid or alkali, depending on what fibre type it's in. If you pre-incubate the muscle section in acid, the type 2 or this fast twitch fibres lose their myosin ATPase activity, so that when you stain for that, you see a dark stain. So on the left-hand side of this, with the acid pre-incubation at 4.6 and staining for myosin ATPase, you can see the type 1 fibres or slow twitch fibres. If you reverse the pre-incubation and do that in alkali and then stain for myosin ATPase in the next section, you can see the reversal of the staining intensity. The fast twitch fibres now stain darkly, and the slow twitch fibres have a less intense stain. And this was the way in which muscle fibre type distributions were measured and estimated in human samples for many years. In recent years, with the development of antibodies to specific proteins in muscle, another approach is to characterise muscle fibres using an immunoblotting approach with specific antibodies. In this image, we have 5 individual single fibres dissected from a muscle sample obtained from a muscle biopsy. They're then blotted with antibodies to either the type 1 myosin heavy chain or the type 2 myosin heavy chain. And you can see these 3 fibres have the type 1 myosin heavy chain, so they're type 1 fibres, and these 2 fibres have the myosin heavy chain 2 protein, and they're type 2 or fast twitch fibres. You can also see the fibre type differences in another important protein that's involved in muscle contraction, troponin C. Troponin C is the protein that interacts with calcium and the actin and myosin filaments within muscle, and you can see that there are fast and slow isoforms of this protein which are expressed in the appropriate fibre type manner. If we characterise the fibre types that are found in human skeletal muscle, as I said, there are 2 main types, type 1 and type 2, and the type 2 can be subdivided into 2A and 2X. It's important to note that the differences between individual fibres in human skeletal muscle are much less than you see in rodents or other experimental animals that have been studied, where there are much larger extremes and bigger differences between the individual fibre types. The table here summarises the differences in characteristics between these various fibre types. The alternate names for type 1 you can see are often referred to as the slow oxidative or the slow twitch. The type 2A, the fast oxidative glycolytic, or type fast twitch A. And in the past, the 2X fibres have often been referred to as fast twitch B. In most limb skeletal muscles in a human, there is no 2B myosin heavy chain. It's found in some animals and in some of the very fast contracting muscles of the eye. But in most limb skeletal muscles, what used to be known as 2B is really the 2X myosin heavy chain isoform. You can see differences in the mechanical properties of these fibre types. The force and power output is higher in the type 2 fibres, and in contrast, the endurance capacity or the fatigability is higher, the fatigability is less in the type 1 fibres. This relates in part to the mitochondrial differences and the supply of blood. So the type 1 fibres are more dependent on the oxidative energy system. The fibre areas, the type 2 fibres tend to be bigger than the type 1 fibres. And if you look at the distribution roughly in an untrained vastus lateralis, a muscle of the thigh, it's usually about 50-50 fast twitch, slow twitch. And obviously there are individual differences. The functional significance of these different fibre types plays out when you look at how they're utilised during exercise. And if we look at how muscle fibres are recruited to contribute to the increased force requirements as we move from light exercise to moderate intensity exercise to very intense exercise, this is really governed by what we know as the size principle. Those motor neurons that are the smallest and innervate the slow twitch fibres are activated first. So at the lower exercise intensities, we have primary reliance on the slow twitch fibres. As we increase the exercise intensity and the force requirements of the activity, we increase the recruitment of slow twitch fibres so that more of those fibres are activated, but then we also increase the recruitment of the 2A fibres initially. And as we approach maximal levels, we start to recruit the 2X fibres, or as in this graph, designated as the 2B fibres. One of the early ways that was utilised to assess the recruitment of fibre types was to measure the glycogen levels. And this was done using the histochemistry, so you would characterise the fibres according to their myosin ATPase using that acid and alkaline pre-incubation. And then you would stain the fibre with a chemical known as PAS, which would stain the glycogen content. And then you could measure that at various times during exercise. And you can see here the change in glycogen within these individual fibre types. You can also note the numbers of fibres that were counted. And what should be readily apparent is how labour intensive this was in the early days. Fortunately, in more recent times, there are computer software packages that scan images and make this a much more efficient task. But what you can see from this rather complex slide is that as you move from low to medium to high, you start to rely more on a greater proportion of the slow twitch fibres and more of the fast twitch fibres. So this, in human exercise physiology, was the method used for many years to see which muscle fibres were recruited during different types of exercise, different modalities, different intensities, different durations. And there's a considerable literature that's looked at muscle fibre recruitment during exercise. Another interesting aspect of muscle fibre types is their relevance to sports performance. Not surprisingly, the suggestion is that endurance athletes who have to exercise for prolonged periods at relatively lower intensities and resist fatigue might have more slow twitch fibres. In contrast, sprinters who have to generate very high forces and only have to do so for a relatively short period might have more fast twitch fibres. There's been a lot of debate as to whether fibre types can be transitioned, change as a result of different types of exercise training. But if you look at the studies that have looked cross-sectionally at the fibre type characteristics of various athletic groups, we do see differences between groups of athletes. Here is a muscle section obtained from the leg muscle, the lower leg muscle of Frank Shorter, who was a very successful marathon runner, an Olympic Games gold and silver medalist in the marathon. And he was studied in the mid-70s by a group of researchers in the United States led by Professor David Kostal. And he's generously supplied this picture, which shows the cross-section of Frank Shorter's muscle pre-incubated in acid so that the slow twitch fibres stain dark. And as you can see here, it's readily apparent that Frank Shorter's muscle had a lot of slow twitch fibres. If you look at fibre type distribution in various athletic groups, and this has been done by a number of investigators in the sort of early to mid-70s, you can see this fibre type distribution. So in untrained subjects, roughly 50% or so of slow twitch fibres. This is higher in the endurance athletes, but lower in the sprinters. So the sprinters have many more fast twitch fibres, and the endurance athletes have many more slow twitch fibres. And one of the questions that has to be asked is, are these athletes born this way and therefore have a natural predisposition for these various athletic disciplines, or as a result of training, have there been adaptations and transformations within muscle that explain this? It's probably a bit of both, although it's thought that there is a large genetic component to fibre type distribution. Having said that, prolonged endurance type exercise has been shown to involve some fibre type switching and transition from 2A fibres towards type 1 fibres, certainly in their metabolic characteristics, if not in terms of their contractile characteristics. An interesting aspect and an interesting study involved looking at different types of athletes in different events. And what they did here was they looked at the slow twitch fibre in a leg muscle, the vastus lateralis, and compared it with an arm muscle, the deltoid. And you can see in runners, they had relatively greater numbers of slow twitch fibres in their legs. On the other hand, kayakers, who heavily involve their upper limb muscles, including the deltoid, had relatively more slow twitch fibres in their deltoid muscles. So this might suggest that over time there are adaptations within various muscle groups that are specifically recruited during specific training that might induce adaptations in fibre types. Equally, you might argue that the success of these particular athletes, either in kayaking or in running, was a consequence of their natural fibre type composition. In our last module in this course, we're going to examine some of the aspects of genes and athletic performance and ponder, are athletes born or are they made? And one of the questions that applies to is clearly muscle fibre type.